Hi everyone, my name is K. Ramnachari, private faculty. Today we are going to discuss about the topic. Today we are going to discuss about the topic DNA. Actually, cell. First of all, cell. Cell discovered by Robert Hooke. That is the non-living cell. Living cells first discovered. Living cells until when living Hooke. And after that, cell mainly majorly three parts are there: plasma membrane, cytoplasm, nucleus. Plasma membrane outer layer, cytoplasm, plasma membrane, nucleus, that is the between the colloidal gel-like structure. Different organelles also present: lysosomes, Golgi complex, mitochondria, different type of organelles. But nucleus is very important controlled cell. The nucleus contain different type of materials are there. The main important first one is DNA is very important genetic material. DNA main important genetic material transfer inheritance character one generation to another generation. Okay, now history first of all in 1869 Meacher discovered nucleon in the cells from pus, and later he spread into a protein and an acid molecule. It came to known as nucleic acid after 1874. That is the of nature. That is the statement of nucleus DNA first time. And 1926 Levene proposed tetranucleotide theory, which states that nucleic acid consists of only four nucleotidases. It gives for different nucleotides and hydrolysis process. That is the main important. History, DNA history. And after that, DNA main important. Genetic material, main important DNA genetic material, all of mostly all different type of organism except some viruses. Next, continue. And after that, Rosalind Franklin used X-ray crystallography to help visualize the structure of DNA. Rosalind Franklin, that is the base DNA structure, very important research. Rosalind Franklin, that is the after this research based Watson and Crick well developed DNA structure. Next, this one is James D. Watson and Crick, James D. Watson and Francis Crick, co-organizers of the double helix model. Double helix DNA, main important to double helix structure DNA. After that, the research they got Nobel Prize. And after that, main important DNA or deoxyribonucleic acid is the hereditary material in human and almost all other organisms, like different type of plant and animals. And after that, nearly every cell in a person's body has the same DNA. Nearly every cell in a person's body. And where is it located? Most DNA is located in the cell nucleus, and where it is called nuclear DNA, but a small amount of DNA can also be found in the mitochondria, where it is called mitochondrial DNA, empty DNA also. And after that, the structure, DNA structure. DNA, the meaning of DNA, the meaning of DNA, deoxyribonucleic acid structure. DNA has three main components. How many components are there? Three main components. First one is deoxyribose, a pentose sugar. Base, there are four different ones. adenine, guanine, cytosine, thymine. This is the base pairs in DNA. But adenine, guanine, cytosine, uracil, that is the RNA base pairs. But the DNA base pairs are adenine, guanine, cytosine, thymine. And after that, phosphate. The main structure, DNA has three main components: deoxyribose and base structures and phosphate group. And structure. The structure of DNA is illustrated by a right-handed double helix with about 10 nucleus spots, main important periodical term. Each spiral strand compared is sugar phosphate backward attached base is connected to the complete hereditary by hydrogen bonds. Main important four parts: adenine, thymosin, guanine, cytosine. Main structure of DNA. This is the model structure. Model structure DNA. The main important JP Watson and Crick, FHC Crick, in 1953 designed the structure of DNA. It is called the Watson and Crick model of DNA. They were awarded with Nobel Prize for this work. According to Watson and Crick, DNA in the form of a double helix. Next, the basis. The bases. There are divided into two groups: pyrimidines and purines. This is a very important, pyrimidines and purines. Pyrimidines made up of one 6-member ring. This is the 1, 2, 3, 4, 5, 6. And pyrimidines thymine and cysteine. Thymine. And after that, purine. Purine made up of 6-member ring fused to a 5-member ring. Adenine, guanine. Adenine, guanine. Cytosine. This is the thymine and cytosine. Pyrimidines and guanine, adenine, and after that, adenine and guanine. This is the purine. The base pairs main important. This very important structure. Chargaff law rule in 1950, Erwin Chargaff formulated important generalization about DNA structure. The generalization are called Chargaff rule. In this honor, they are summarized below. Main important summarized: a Chargaff rule may be 1:1 ratio, that is the adenine plus guanine, that is equal to thymine plus cytosine. That is the Chargaff law. In 1953, Watson and Crick postulated a 3-dimensional model of DNA structure that accounted for both the X-ray data and the characteristic base pairing in DNA. It consists of two helical polynucleotide chains. Two polynucleotide chains coil around the axis, a form of right-handed double helix. In the helix, the DNA2 chains are strand or anti-parallel, have an opposite polarity. Backbone of each chain, which consists of alternate sugar phosphate residues, main important are on the side of the facing the surrounding. And after that, this is the DNA detailed study. The purine and pyrimidine base of each strand free inward towards each other. The bases are stacked perpendicular to the long axis of the double helix. The base pair are 0.34 mm apart in DNA. Helix, a complete turn helix takes 3.4. Therefore, in each helix turn, 10 bases are present. The external diameter of helix is 2 nm. building small or finally small molecules are reached. The functions of the function of DNA. Nucleotides are precursors of the nucleic acid, deoxyribonucleic acid, and ribonucleic acid. The nucleic acids are concerned with the storage and transfer of the genetic information. Genetic different type of plants, different type of animals transfer genetic information. Actually, main base DNA formed double stranded DNA formed single stranded RNA, especially mRNA. That one is called transcription. That is the mRNA transfer to protein. That one is translation. That main based this DNA important role. The universal currency of energy, namely ATP, is nucleotide derivable nucleotides and are also components of important coenzymes like NDA plus and FAD and metabolic regulators such as CAMP and CGMP. DNA must be stable. DNA every time must be stable. There are coding and non-coding regions found on DNA, and coding regions code for genes. There is the proteins. Non-coding regulation can be either DNA junk or help regulate protein synthesis. Now main important now completed what is the DNA, deoxyribonucleic acid. DNA main important through three parts: sugar, nitrogenous bases, and phosphoric acid. What is the sugars? What is the nitrogen bases? Adenine, guanine, cytosine, thymine, and phosphoric acid. Structure of DNA, Watson quick rule, and Chargaff law will be Chargaff law completed. Now coming topic of DNA replication. Replication main important to DNA. Replication is the duplication process by which a DNA molecule produces extra copies of its own structure. Replication occurs inside the chromosomes. It occurs during interphase. The parent DNA strand function as templates for the synthesis of new DNA strand. New DNA is produced by semi-conservative process from the strands produced. One strand is the parent strand and the second strand is the new synthesis strand. DNA replication required the following components: DNA templates, a primer, DNA, DATP, DGDP, DTDP, DCTP, MG++, DNA, unbinding protein, DNA polymerase, RNA polymerase, ligase, deoxyribonuclease. Main different type of. The type of DNA replication: semi-conservative model of DNA replication in prokaryote DNA replication, eukaryote. DNA damage, types of agents of mutations, spontaneous radiation and chemicals. Repair mechanisms and DNA combinations. Main important to central dogma: replication, transcription, translation. But this is a DNA double helix DNA formed single stranded formed RNA. That's one is the transcription. That's our single stranded RNA formed protein. Translation process. That is the central dogma. Main transcription translation process: initiation, elongation, termination. Also very important. DNA, RNA, protein. This is a reverse transcription. Reverse. This is a RNA to DNA. Transcription DNA to RNA. This is a very important central code. Types of DNA replication: main three parts: semi-conservative DNA replication, conservative DNA replication, dispersive DNA replication. That is the main important. First of all, semi-conservative DNA replication, conservative DNA replication, and dispersive DNA replication. Semi-conservative DNA replication. The method of DNA replication was proposed by Watson and Crick. In this method, one strand of DNA molecule serves as a template for a synthesis of new complementary chain. So during the replication, one strand is the parent molecule is incorporated in the daughter molecules. Another strand is parent molecules. Main important this method is semi-conservative method. And main important this proposed by Watson and Crick. This semi-conservative method, this is the main important two standards. But this one standard is mother standard, one standard is daughter standard. That one is semi, indicates semi-conservative DNA replication. Next, conservative DNA replication. The method was proposed by Cavalieri and Rosenberg. According to the method, the DNA strands do not separate. The two strands template is produce a new daughter double strand. Thus, of the two double helix formed, one would be entirely of old material and the other double helix is entirely a new material. This is a conservative DNA replication. The mother strand, totally this is the two strands are mother, two strands are newly. This is the conservative DNA replication. And dispersive DNA replication, final one. Dispersive. According to this method, the parent strand molecules break down to the small pieces. Each piece synthesis a small DNA molecule. Then the daughter DNA molecule is assembled by the linking of the old and new pieces at random. Yet the pieces of parent DNA molecule remain scattered the daughter DNA molecule. Okay, everyone. This is today topic. What is the DNA? What is the DNA structure? Function of DNA, DNA replication, three parts: conservative DNA replication, semi-conservative, dispersive DNA replication. Thank you so much. Do like and subscribe my channel. K. S. Ramnachari, Ideas.